All right, I think we can go ahead and just get sort of the introduction. We are so excited to have Bogdan Pasaniuc. How do I say your last name? Pasaniuc?
Yeah.
Okay, close enough. Who is new, not a new professor, but new to Penn. So how long have you been here now?
3 months.
3 months in the Department of Genetics and affiliated with the Institute of Biomedical Informatics, where he is running a center called
Equitable on the spot. So Center for Computational Biomedicine.
Center for Computational Biomedicine. He comes here from UCLA most recently. And I think I'm just going to keep this really short and let you tell people what your interests are. But certainly you're well known for doing a lot of the work looking at admixture and trying to have more effective polygenic scores for people of diverse ancestry. So we're looking forward to hearing your talk.
Great. Thank you so much. So I don't know if I have to speak in this microphone. Does it work without the microphone? I'll try to speak louder because otherwise. I'm sorry?
Oh, that's how they hear online. Yeah.
Oh, online goes through the microphone. Okay. All right. I'll try to be close. I guess if I'll move or I'll try to stay here then. So things work out. All right. So today I'll be talking to you, or thank you so much for inviting me here. It's really exciting to meet new faces and see familiar faces as well. If you guys can hear us over Zoom, just type it there, and I'll try to get back to the microphone. So today I'll be talking to you about the equitable polygenic risk score for precision medicine, and really, I'll dive right into it in the interest of time because I'm really interested about how you guys think about this, and please view this as an invitation to collaborate in various different domains here. I see a lot of my students are somewhere over there. I'll put them on the spot. So, if I can't answer some questions, they're definitely much more knowledgeable than I am in specific things. Perfect. Oh, there you go. Thank you so much. It's not like we planned it, right? So, Jinja, do you mind standing up and introducing yourself? Maybe like one sentence.
Yeah. Glad to meet you all. And I follow Boston Code from Seattle. One of the few that work on single cell. Yeah, it's really fun.
Welcome, and so sorry about the weather, but
It's only going to get worse.
it's definitely going to get worse. Yeah.
Sandra, do you want to say a few words? Maybe like one sentence. Great. Alex?
Hi, I'm Alex. I work with polygenic scores for autism and looking at advocacy.
Hello everyone, I'm Bogdan. I love the weather.
Okay, but not just the East Coast.
Yeah. Yeah. Also moved from UCLA, and I work in admixture, PRS, and some people.
Great. And then there's another person that's not here, Ravi, who's decided to take a vacation before the holiday. So just not to be put on spot by Maya, I guess. So today I'll be talking to you about polygenic risk scores, and I'll please view it, like I was saying before, as an invitation to work together on the various different areas of here, because I know that there's a lot of expertise in this room and beyond. And really I wanted to put Sarah on the spot also in here, and really this is a year-long talk in the making. So this talk I've been thinking about for like a year or so, and kind of like the way when Sarah asked me a couple of weeks ago to give this talk, can think about it as an update to the answer to this question. So a year or so ago, I gave a talk also about polygenic risk scores here at Penn. It was over Zoom and whatnot. And as I usually give these talks, I realize that I tell a lot of the bad things about polygenic risk scores, why they don't work, why they have all these issues about equity, accuracy, interpretability, and so on and so forth. And at the end of the talk, Sarah asked a fantastic question, which I don't know if I gave a right answer or a good answer at the time. So this is paraphrasing. All these words are not exactly your words because I don't remember word by word. But the question was, is there anything salvageable from this? Is there anything that we can actually do useful with polygenic risk scores in the long run? And I think at the time, I may have provided a good enough of an answer, but hopefully in this talk, I'll try to convince you that not only that they're salvageable, but we can do a lot of amazing things with polygenic risk scores. But obviously, we have to not go into this other domain of overhyping and trying to pretend like polygenic risk scores solve everything, but actually take it, equitable, if you will, or all these different kind of concepts and actually solve them methodologically before we actually implement them. So the talk today will be about different areas of polygenic risk scores, how they fail, if you will, and how do we use methodology. So my lab is a lab that runs computational and statistical methodologies. How do we potentially fix some of those as we propagate them through the clinic in medicine in general? So I'll summarize in 2 slides 20, 30 years of discoveries in genetics, if you will, even not more than 20, 30 years, that fueled the era of polygenic risk scores. So one of the big summary, and this is tracing back hundreds of years ago, but more and more recently, it's kind of more sharply fine-tuned, it's that everything that we measure in humans has a genetic component. It's not genetically determined, because genetics is just one factor of many different things, but really every phenotype, every trait, every disease that we measure in humans has a genetic component. And there's many different ways that you can make that statement. I really like this work from 5, 6 years ago that essentially looked at lots and lots of traits, as many as they could measure, 20,000 in that case. And this is a histogram of the correlation between twins, identical versus fraternal. And what's really interesting here on this figure is that all these densities are shifted away from 0. So they're like on the 0.5 and closer to 1, which says that the more genetics you share, the more of a genome you share, the more similar you are at the level of phenotypes or tree, right? So if we think about medicine or implementing, let's say genetics in medical domain, it's great because everything that we measure in all of our patients has a genetic component, right? That again doesn't mean that they're genetically determined. So that means like if you have if the genetic predisposes you to have a disease that you're going to get it, but the genetics is one of the many components of it. And the other big finding, in my opinion, from the past 20 or so years of large-scale genome-wide association studies, actually more than 20 years, ever since Neil Risch wrote that fantastic paper on how do we actually do it, like a decade before GWAS actually did it, and figure out a way to map lots and lots of regions in the genome that are essentially containing risk genes or risk variants for complex traits. And the main finding is that every common disease in humans is highly, highly polygenic, right? And this was a matter that was highly debated, 10, 15 years ago. There was like massive debates in the field about whether, a trait like, let's say, like heart disease would be, the genetics of it would be kind of characterized by, let's say, a dozen or so key core genes, whatever we want to define them, and, a tail of like less important genes. So that was one of the side of the debate. The other side of the debate was that virtually it's highly, many, many genes contribute to heart disease, and it's going to be kind of in this hard to tell apart which ones are core and which ones are not. So the long story short is that for a individual, which is displayed in this karyogram, to be high risk for heart disease, it's an aggregation of lots and lots of variants or risk variants that each of them has a tiny little contribution, and that puts that particular individual in the high risk category, right? And these 2 findings, which I highlight on for the past, let's say, 20 years or so, really set the field for why do we even bother about aggregating the effects into this simplest way that we could do machine learning or prediction by just summing all these things up. So not too sophisticated of machine learning models, just summing up all these variants, and we can essentially put some weights on top of them, which depends on the effect size, and that gives us this idea of polygenic risk. All right. So that's my intro slides. I'll stop there. If you guys have any questions, I'm not going to answer them. There's reviews that you can read, and I can point you to the literature. But the promise of how do we want to use these in the medical domain, in clinic, is that whenever we see a patient, or whenever I go to, or you guys will go to your clinician will already have your genetic profile and already will be able to put you on these distributions for all these different diseases and kind of tell whether you're a high risk or low risk for various different conditions. You might, in this case, what's listed here is cancer, heart disease, asthma, and so on and so forth, right? So I purposely chose common diseases, but obviously it's already happening for rare diseases. It's just that very few people carry rare diseases. So in this, at core of this exercise is this idea that there is a distribution that most of the time looks normal because everything looks like a Gaussian or bell shape. And then somewhere there's a threshold for whatever the clinician may want to do with you. Let's say prescribe statins, for example, if you're a high risk. So that's where that dotted line, red line stands for. And then there's a statistical underpinning of this, which means of how do you define whether a particular individual is high risk or low risk? How do you put someone above that threshold? Right? And this is really what I call the N of 1 problem, which is, I think it's a different way to think about polygenic risk scores. And I'll try to convince you in the next 40 minutes or so that by thinking about it in this way, one individual at a time, we have new sets of problems and methods to solve. Okay, which gets back to Sarah's original question that motivated parts of this talk. So just to scare it straight a little bit, this already happened. So, a couple of years ago, as we are talking about polygenic risk scores, we're kind of imagining this future where genetics or genomics will just go through the clinic and will be used. As it happens, most of the times, there are tons of different startup companies that are already, in many different ways misusing this technology, but they're selling these products that purposely do all sorts of different things from, doing fun things like measuring your genetic ancestries, and I use quotation here, what it means to be fun there, versus, crazy things like, using polygenic risk scores to figure out how you design your babies, if you will, right? And again, this is like to scare you straight that there's a role for all of us to figure out what are the limits and where's the science ending and when the snake juice kind of like salesman starts. And interestingly enough, you see companies nowadays that are selling polygenic risk scores as a tool for when you get insured by those companies. And if you went to ASHG this year, you probably have seen some talks about, using polygenic risk scores to measure the cost that you as a patient will incur to the medical enterprise, right? So you can imagine where would those things be misused, right? If there's a genetic score or that predicts how much you're going to be, the medical enterprise is going to pay for you on your behalf. We can imagine how that will play into issues about equity, who gets access to care, and so on and so forth. And more surprising, all these researchers are coming from Europe, where there's universal pay, so there's not that much of a concern, if you will. But when they come here in the US, you can imagine all the crazy that's going to happen. So this is just a scary straight to get here. If this talk would be in the morning, you'd be kind of waking up a little bit, but hopefully we can start to solve some of them. So one of the big challenges that's coming now in the field, and this is the part of my talk where I tell you the challenges and then we can try to solve them, is this idea, and this is happening more and more nowadays, and this idea that you take the same patient, the same person, you try to measure whether that person is a high risk or not based on polygenic risk scores, and for the same disease, and based on what polygenic score you pick, you get different estimates, right? And for example, in this talk here on the left from David Hunter in the UK, they looked at cancers and they found that, only, a very small amount of people that are high risk, they're consistently labeled as high risk based on polygenic risk. And that's true throughout. And from our colleagues here from Penn, they published a paper in JAMA yesterday or 2 days ago that made this point even more thoroughly looking at heart disease. So here on the left side, you see from their publication, and on the right side, you see the citations and the editorial comment. So on the left side here, you see 3 example participants or patients, if you will. And then on the columns there, they're different polygenic risk. And they're ordered by year because, they have to order them by some. And what you see there in the plot is the percentile for that particular patient to be high risk or low risk as a function of, where that polygenic risk score was at. So if you take the participant 1 or individual 1, you see that, that person sits at the 50, 60 percentile, drops to close to 0 percentile, goes up to 70 percentile, and so on and so forth.
What's changing every year? What is it that's changing every year?
So what's changing here, these are different polygenic risk.
Oh,
So if
okay,
you
so
go
different years, they use different polygenic risk scores.
different years, there's yet another new polygenic risk score that gets developed and then that gets published somewhere.
So like they went back in time, looked at what was available in that year or
Mm-hmm.
something, ran it on the patient.
Exactly.
rated it for-
That's exactly what it is. So here's 50 or so polygenic risk score for heart disease. They're ordered by the time of publication. And then you go back to the same patient, participant one, right? Obviously, their genetics doesn't change, right? We're talking about common variants. So genetics doesn't change. Every year, some researchers publishes a new polygenic risk score, and then you start seeing this particular person, high risk, low risk, all over the place, right? And this particular, that's why they chose this participant, right? Because it fluctuates all over the place. But you can see like this guy here is, kind of highest risk or like some medium risk, high risk, again, medium risk, and so on and so forth. So the big problem that they're emphasizing in that paper is that you cannot really do anything meaningful in a clinical sense with a predictor that's all over the place, right? Because the genetic risk should be fixed. You're trying to predict something that it's a fixed quantity, but you're jumping all over the place based on whoever designs whatever polygenic risk scores that they do.
Oh, it does look like if you combine them all together and then look at the one on the right, does that give you some information?
That's a great idea, and that's exactly what this paper is trying to do. So if this paper from Perdina Berenjan from Harvard, they're proposing something like this, where it's like you average things out and then you get something that is more stable, right? The challenge there is that whenever you average things out, you get the average, which is supposed to be more stable. So you don't know if it's stable because you're just averaging a bunch of you're getting closer to the ground truth, or you're just getting something stable because you're just averaging a bunch of numbers. Right? So, and I'll get back to it in maybe 5-10 minutes. And kind of like my message to you today is that you'll start seeing a lot of these types of reports because they mirror what happened a decade or so ago with genetic ancestors. For those of you that are more junior in the room, 10, 15 years ago, there used to be a large number of papers or journal articles in New York Times and all this on all the media, where people would just take their DNA, or they would spit it in a tube, they would send it to 23andMe, they would send it to Ancestry.com, they would send it and they would get different answers. So one company would tell them that they're, this much percent European, the other company would be telling them they're other percent European, and they would essentially question the utility and robustness of all these measurements. So my prediction is that in the next couple of years, you'll see similar types of journal articles that will come up about polygenic risk scores, common diseases, and so on. So it's up to us, the scientists, to try to solve them or try to understand why that occurs in the first place. So in the first part of the talk, I'll try to convince you that this is fully expected. I'll try to convince you that this is just statistics 101. So once you understand the stats 101 of polygenic risk scores, it makes a lot of sense why they jump all over back and forth. And hopefully I'll try to convince. If not, please, let's work together. Maybe we'll find a way to convince each other. So I only have like a few slides of math in this talk. So, this is a few of them, but happy to have more math and more mathy conversations, but I didn't want to lose you into this. So what is a polygenic risk? What are we trying to do? So we're trying to get a lot of data. So here in this case, you can think about genomes on the rows here, or genetic data from lots of individuals. And we measure them on lots of sites in the genome. And typically we'll look at common variants. So common variants are on the order of like a couple of million, give or take, doesn't really matter. But essentially there's a mismatch between how many data points we have, so how many individuals with disease status that we have, which is by the largest data sets nowadays are on the order of millions, and then number of features, so number of genotypes is an order of magnitude larger. So we measure this, we measure the disease status or whatever we want to predict, heart disease or not, and we're trying to do a prediction, right? And at the core of the problem is the fact that we don't know the causal mechanism of the disease. So we're letting the machine learning or the model infer that. And typically we do it by just a very trivial or standard linear model that says that the heart disease, or let's say quantitative phenotype, is, you can write it as a summation of genotype effects times the weights. Those are the true causal effects there. And under that type of model, where there is such a quantity, which you can, sometimes you see it in the literature as referred to as the true polygenic risk score, or if you read, or if you take quantitative population genetics, they refer to as genetic value or breeding value, especially folks come from agriculture. If you read this book, which is like, you probably see it's 1998, and that's like a version 6 or so. This is like the so-called Bible of statistical quantitative genetics by Lynch and Walsh. There's an entire chapter about polygenic risk that talks about what I just showed you before, that clinicians are now discovering. It's just that they use these terms that are a little bit of, not a little bit, a lot offensive for human geneticists, that human geneticists rarely read that chapter. They call breeding values and so on and so forth, right? So this is the quantity that a polygenic risk score is trying to estimate, and by virtue of Bayes statistics 101, this is the estimator, that's the estimate, right? And that's why it has the same mathematical form. The only thing that changes there is that those betas are hats. So we're estimating those. Nobody gives us the true causal effect, right? Make sense so far? Hopefully I didn't lose. So by just this very simple statement, I can already hopefully convince you that because we're estimating a value, so it's not really a score when you think about polygenic risk. It actually has a statistical meaning that you can say that it has a bias and a potential standard error. So in other words, whenever you're predicting the phenotype of a particular individual through a polygenic risk score, you have a potential for a bias and a standard error attached. And this is why we see those issues reported before. And without going into lots of details, we wrote a lot of papers about this. I'm happy to dive into the details of this. The crux of it is that we don't know which variants are causals, and we don't have their causal effects. So we don't know the causal methods, which means that we're estimating them from the data, and LD, or correlations between variants, cause a huge problem for us when we estimate. And so here's a very simple toy example in which, let's say, we might see, when we run an association study, we might see similar effect size at these 3 particular genes or variants, let's say. And let's say that the second and third variant are highly correlated among themselves, so strong LD. Then essentially these 3 causal configurations are all equally likely given the data, right? And if you think about a population or estimating in a particular population of individuals, it doesn't really matter whether you pick the first 2 variants to have large effect size when you do a prediction, or the first and the third, or you just distribute the effect among the last, because you're still predicting the same amount of variance in a population level sense. However, for any particular individual, and anytime when you realize that prediction, that causes massive, or potentially causes lots of problems. Because if you have an individual that just happens to have a risk allele at the second gene here, or the second variant, all of a sudden, in this case, you made that individual as high risk. In this case, you made this individual as medium risk. And in this case, you made this individual somewhere in the middle, right? Because you've chosen this causal configuration or the method has chosen these causal configurations pretty much at random. That's like that's what because they all optimize the same. They all give you the same population level performance. Okay. And this is really what's happening under the hood. That's why. So this is an example of 3 variants. And I imagine you have 60,000 or so LD blocks in the human genome. Each LD block has thousands of variants that are common. And this is happening on that magnitude, on that scale, right? So you have tens of thousands of these types of small little decisions that your machine learning or the method that builds out the polygenic risk score is doing. And at the end, just picks one of these 3, and you don't really know why pick that one and what's going to be what's going to be the impact when you actually realize this. So one of the first questions that we asked a couple of years back is whether we can estimate the standard error or the variance in our prediction. I'm not going to go to the details of this, but essentially, you can actually do it with reasonable amount of accuracy under very simple assumptions. And at the end of it, what we found when we applied to the real data, we found exactly what clinicians are now reporting. That if you look at the tails of this distribution, the standard error are so high that any particular realization of a polygenic risk score, it's pretty much kind of random whether you put that particular person above or below the threshold because the standard errors are so large. And that's what this is trying to say. So it's saying that if you look at the full credible interval or the full estimate plus the standard errors, and you're asking whether the entire confidence interval is in the high risk category, then you divide by the total, you get these very tiny little numbers, which means like very few individuals you have full credible confidence that they are truly in the high risk category, right? And another way to kind of present this is to say that now every prediction has a standard error, and that standard error has a meaning attached to it. And you might have this very interesting scenarios in which you have different individuals, their actual absolute risk or relative risk might be different, but also their confidence on what you call above that threshold could be different, right? And we're arguing, or I'm going to be arguing for the next 30 minutes or more if you let me, that these confidence intervals are super useful for bringing equity to the polygenic risk, right? So this is the framework or how we're going to operate with the standard errors. And now let me tell you about the equity part. So what is one of the big sources that drive this answer, right? Obviously, you can into it, and probably that's what a lot of reasons why all of you worked in this space probably, is that genetic ancestries are one of the main drivers of polygenic risk answer, right? So these standard errors are essentially larger for individuals of certain genetic ancestries than other individuals. Why? Because the training data has a mismatch to the testing data. We train it in particular individuals of certain genetic ancestries, and then we apply it to individuals of other genetic ancestries, right? And when I say this, probably the most famous figure in the past 5-10 years is this figure popularized by Alicia Martin and her group in a couple of papers there that highlight the fact that if you train one of these polygenic risk scores in Europeans, so in this population here, and then you start applying in populations that are more genetically dissimilar to that population group, then you see this decay in performance, right? This has been labeled as the lack of portability of polygenic risk. And note that, Alicia did a very wonderful figure here, but there's a lot of things that are happening with it. The more you look at it, the more we looked at it a couple of years back, the more it didn't make sense. So, know that this is 100%. That doesn't mean like you're predicting everything. It's all relative to the performance in Europeans. So the performance in Europeans could still be like a couple of percent, but because you're normalizing by that, you get this kind of like false impression that it works really well in Europeans. It doesn't, right? The other part, which is the part that I'm going to show you in the next couple of slides, is what does it mean to be American? So what does it mean to be African in this case, right? Is this like African individuals from Africa, or is this African Americans that live in, right? Or what does it mean American, right? Where do I put myself? Like I'm American, but I probably would put myself here, right? So over.
Is that what she meant? I always assumed it was Native American. Then I was like, she was doing a lot worse. So she literally just meant like US population.
I think this is like a proxy for this like syntagm of mixed American, ADM, as people.
That would explain that.
So it appears like Hispanic Latino would be most of the category. But this underscores a much bigger, problem here, which is these labelings are essentially kind of abstractions that we or scientists put on top of the data that may not match with how people identify as, or may not match with what we know about genetic ancestors. And when we started thinking about it, because we're methods people, started thinking about what happens under the hood. So under the hood, what Alicia and everyone else, including us, were doing at this time, was that there is such a thing as a genetic ancestry continuum, which is something that people were kind of were writing about all the way from Cavalli Sforza in the 1970s and even before that. But this idea that the genetic ancestry kind of fluctuates across a continuum because allele frequencies change across, space, across time as well, but we're talking about space. There's such a thing as like some decomposition about some gradients. In this case, PCAs is probably the easiest one, but you don't have to think about PCAs. And then on top of this, so every dot here is one person. And then on top of this ancestry continuum, for a variety of reasons, most of it because we have never really sampled everything. So we sample here, we sample there. We get this biased view that, clusters exist, where we impose clusters on top of this continuum. And after we impose these clusters, then we start thinking about, oh, we train most of our models here. Now we apply them here. So now let's say that this is going to be 100% accuracy. It's going to be 75% accuracy. And you can imagine that this is a very poor representation of what do we actually do in practice, all the individuals, right? Make sense? Any questions? Yes. South Asians in the Hispanic cluster. Could be, yeah. So this is just this is not real. This is just me toying around. Might be like the dots might be from some real data, but like obviously the coloring that I just. But yeah, you could be right. Because like your final could be on that. So one thing that coming from LA, one thing that kind of pushed us to think more about it is that whenever we saw in our biobank, many of our individuals fit into this like in between these clusters, and we can never really put them in all these bins. And this is my figure that I kind of relate to my kids. My kids kind of like described all these folks here, and all of them have this feature that they have recent ancestry from multiple of these so-called homogeneous continental ancestry groups, and you can never really put them into this bins. And before you start.
In LA, you guys only look at celebrities.
Of course, that's exactly what it is. That's exactly what it is. You got the right message. And maybe that will be like the snapshot for my next talk in a year from now. So before you start thinking about it as like this is a rare thing, we shouldn't think about it. Well, it turns out that 1 in 20 people nowadays, and you can argue that this is only going to increase, fit that category. So the category of like, you cannot really, but based on like these broad categories of ancestry, you cannot really fit them because truly genetic ancestry sits on a continuum. So anytime you draw some circles there, you will make some essentially you'd exclude some individuals, and then you include some individuals in categories that should not be included. Right? So this is like the numbers. Atlas is the biobank at UCLA. I'm assuming that if you do this exercise here in Penn Medicine, you'll see similar type of numbers. And then the other part that kind of motivated us to drop the clustering altogether is we started looking more thoroughly in each of these ancestry bits, right? So obviously this is like just 2 dimensions to explain ancestry across, let's say, at a global level. Once you start looking at each of these continental ancestries, you start seeing more cluster, right? Or more structure, I should say. So here's what happens if you take this East Asian American cluster and you run another PC just in that cluster and you replot them. And because these are patients within within UCLA, we have their self-reported essential ancestry. And if you essentially relabel individuals based on what their language is at home, you start seeing other structures. And I suspect that if you take let's say all the individuals identified as Chinese, you run another PC there, you would see more structure. It's just a matter of like how much data you have, and ultimately there's no cluster that will be a good representation of all this data. So obviously this is not news for any of you, probably. This has like been reported multiple different times. This is just the recent amount of work that has been showing this. I really like this work from Anna Lewis and colleagues that highlight this is the biobank in Mount Sinai, New York City, and every grade out there is one individual. And this is for reference panels from all these different parts of the world. You can start seeing, I don't know why this moves faster, but you can start seeing how genetic ancestry sits on a continuum, and the fact that we sampled just parts of it gives this bad representation that we're in a cluster mode. And every time that I talk about this, inevitably, the question is about how about all of us? And all of us chose, a fancy statistical technique to plot the genetic ancestry. So this is data from all of us from their first 200,000. And instead of looking at PCA like most folks did, they looked at the UMAP space. And obviously, UMAP creates clusters because that's what UMAP is designed to do. So it gives this very poor representation of genetic ancestry as a clusters because UMAP should not be used for genetic ancestry inference as of now, especially when PCA is so well characterized. So this is essentially the first 200,000, and they plug in this UMAP space, which kind of forces clusters on the data, and then they recolor this based on race, and you get this biased view, in my opinion, that you cannot essentially map race into genetic ancestry cluster, which I don't think it's correct. It's more precise to look at it this way. Hopefully that makes sense. And I can say more bad things about all of us first, right? So our idea was what happens if we drop the clustering altogether? So let's ignore clustering. Let's start thinking about in population genetic terms about a continuum of genetic ancestry. And let's come up with, let's say, can we characterize the drop in performance by looking at a continuous measure of ancestry, right? So if you think about FST or like some difference in allele frequencies across a gradient, you can also kind of extend that to one genome at a time. And imagine that you might have some training data where you test these polygenic risk scores. There's a new individual that may not be from the training data, but sits at some genetic units away from that, right? And again, for those that have taken PopGen, this is like FST, right? But on a more one individual at a time. And the hypothesis that we had in that work was, if I have this distance and then I come up with a measure of uncertainty or error by individual, and now I replot the accuracy one individual at a time, the big question for us was, will I see clusters, like Alicia's work was saying, or would I see a gradient of decay? Make sense? So that was the hypothesis at the time. And obviously I'm showing you this because that's what we saw. But before I show you what we saw, essentially we can actually estimate this. And I have these for those that are more mathematically inclined, but I'm going to skip over that. So essentially you can estimate the uncertainty or the error one individual at a time. How you do it, happy to talk to you about. But when we actually applied it in UK Biobank, where you look at, there's a bunch of data of individuals that are within the UK, but have genetic ancestries from other parts of the world. For example, from other parts in Europe, Poland, Italy, and going all the way to other parts of genetic ancestries. When you start seeing the individual polygenic risk or accuracy, this is in simulations, we start seeing this linear decay, right? And that's matched by essentially real data observations where you could actually bin individuals by genetic distance and then correlate the phenotype with the polygenic risk scores, then you see a similar type of trend. And the punchline is that when we did that experiment, this is from 30,000 people at UCLA, we start seeing essentially a very linear gradient of decay in performance. So you start seeing, so every dot here is one individual, and you can already start seeing that the clustering is kind of like a bad summarization of this fit, right? You see individuals kind of like following this gradient of distance, which is similar to what I showed you in the PC plots, and then the accuracy decays linearly. And in fact, if you just recolor all the individuals based on those genetically inferred ancestries, you start seeing how strongly they overlap, right? And in terms of equity, there isn't a population that it's essentially different than others. All populations are the same. There's in all the types of genetic ancestry groupings, we see the same type of structure that correlates with polygenic risk. So even if you test it just in the individuals that are labeled as European American based on genetic ancestry inference, you still see a decay because individuals that have European American genetic ancestry would still sit at different levels of genetic distance from the training data, which in this case was UK, right? And you can see how it varies across. And those folks on the end are the ones that we started this exercise, the so-called unclassified, or people that would be not part of any of these studies.
Those just highly mixed or something?
Yeah, so they're multiplied, so it makes, so this is like, that makes that more traditional populations, Hispanic Latino Americans, African Americans. These are more individuals that have, let's say, one grandparent from Asia, one grandparent from Europe, one other grandparent from somewhere else, and you cannot really fit them into any of these. So, yeah.
So how much, for a given genetic distance, does it differ? How well is that capturing all the decay?
Yeah, that's a great question. So, and it captures 80–90% of it, and then the rest is kind of like a population specific type of a term, which is what you can see here, but the slope here is kind of like population specific. So we think that there's a big part that is linear, and then there's like a lot of room for GYE or like other places. Yes, this is explained there. Yeah. And now, because at the time there were a lot of polygenic risk scores that were used in the clinic for, let's say, Europeans, but not for, let's say, Hispanic Latino individuals, because they were concerned about being inaccurate. Well, it's like there are classes of individuals within this group. So you can put an accuracy here, which would be like, the rationale for why you would use or not use a polygenic risk score. You'll start finding classes of individuals that have the same accuracy of Europeans. So the justification of not providing a polygenic risk score for that entire population is not it's unjustified, or it's not a good criteria. All right, so ancestry impact polygenic risk score interpretation. So these standard errors that I started with that will magically solve all of our problems will have to be varied across individuals given on ancestry. So what I showed you before, the individual might have more European genetic ancestry, might be more precise in our prediction than individual that it's less, right? And I would argue, and you could contradict me, but I would argue that within the past 5–10 years, we kind of solved this. So we know that we need more diverse data, but we kind of know based on, like theory and everything, what's supposed to be happening. So obviously we need more diverse data, but on the method side, we know exactly that if you're sitting further away in units of genetic distance, you're supposed to be lower accuracy. We can model those, we can project all of those. Right? So we have a really good understanding, I would argue, of the impact of genetic ancestry on prediction, on polygenic risk of prediction. And I argue that's one of the big positives in the past 5–6 years.
Well, within a certain environment, so like these are typically all US, for example. We do not yet know people in very different environments and different ancestries, right?
That's right.
I mean, that can have a big role.
That's right.
So
Which
Africa, we try to apply this, and depending where in Africa and the groups and everything, there could be much more variability there.
Yeah, that's all right. So this is all within a similar context/environment. And before you start thinking that Sarah and I, we planned this, that's exactly what I think the next big step is, which we don't know. Yes. So the next big step is, and I call it context here, because I want to include not just environment. So environment, I want to include stuff that could also be genetics, like other comorbidities, other cofactors. For example, heart disease, BMI. Should BMI be a context? Maybe yes. Maybe you should consider BMI, right? Or age or sex and so on and so forth. So I call them context, but I'm happy to change my impact. I'm thinking about environment. So, like what I was saying, whenever there's a big discrepancy between when we apply these versus when we train these. And ancestry is one of them, and we kind of understand what it is, but in general, the context or environment is way different. Here we train them, let's say, in very well-defined cohort or biobanks where we minimize all sources of heterogeneity. Environment is one of the most rigorous one that we try to account for, because we try to avoid false positives as much as we can. But then when we go out to try to implement this, we took data from UK and we applied it in the US. We take data from the US and we try to apply it in Africa. And obviously, environments are different, but even within the US, right? We're trying to apply it to Penn medicine, but all these predictors are coming from, let's say, all of us, right? So there's a big discrepancy between the two. And in my mind, one of the first papers that kind of like showcases this systematic problem, there are many others, but like this paper from Ole Prashovsky's group from Mostasavi et al. Is showing you that it doesn't really matter how you train and how you account for these contexts, you're going to have differential predictions. So this is, if you're predicting blood pressure, you see you have a differential performance men versus women. If you're trying to predict BMI, you're going to be more accurate in young versus old. If you're trying to predict years of schooling, for whatever reason you may want to do that, you'd see that it's differential performance based on socioeconomic status, right? So these are different contexts that will impact the way you think about, those standard errors that will come into play. So when we looked at the data, read that paper, Kang Chan, a grad student in the lab at the time, was trying to think of in a critical way whether Molly's group cherry-picked a couple of these traits to showcase, kind of like, or it's a pervasive type of effect. So what he did, he looked in the Biobank and all of us in the hopes of answering the question, is this happening a lot, or is just, for example, true? And as you might expect, it happens a lot. So I don't have time to go into all the details here. You can read more about this paper that just came out a couple of months ago, but just picking LDL as an example. So this is like this column here. These are different contexts, age, sex. This is genetic ancestries that's coming up from PCs. BMI is a context, smoking, whether you use alcohol based on self-reported in the biobank, whether you wear glasses or not, which is a fun context to think about. Income, not a fun context to think about. Deprivation index, the same, or education index, right? And if you just look at this, what Kang Chan did, looked at the tails of the distribution of the context. So, in this particular case, you're more accurate in the young than in the old. You're more accurate than whether you're a woman than a man, PCs, and so on and so forth. And interestingly, you're more accurate whether you not wear glasses than whether you wear glasses, right?
Maybe tracking socioeconomic status?
It tracks age.
Oh, age.
It's simpler than that. So if you account for age, that goes away. But that's a fun thing to think about for us from the methods perspective, that emphasize that we should consider all these contexts together. And I'll get back to it in a second. This is UK Biobank, and this is like reflecting what I think Sarah may be hinting at, which is that this is a very well-controlled resource. They purposefully selected people from a particular health system or particular centers, if you will. They're older, they have like a particular composition. If you go into a data set that is more diverse, like all of us, you see a much bigger impact of the context on prediction of the polygenic risk, right? And again, don't have time to go into all the details, but hopefully you can agree that the color scheme is more thorough there than on the other one. So the other conclusion here is that now we have to worry about context as well. Whenever we predict things, we don't only think about genetic ancestries, which is, like I was saying, it's an easy thing to think about because the further you away, the more inaccurate you are. In the context space, it could go all the different ways. And it could go it could be more accurate, less accurate. You have to actually think about one disease at a time. So how do we go about doing that? And this is the part where I'm hinting towards solution, and I realize I don't have much time, but the way we think about it is to actually calibrate these models in real medical systems before we actually use them. So the idea being that instead of going through directly from a training model towards the data, you have this magical data that sits in the middle that happens to have the same representation as the data that you're trying to apply for into. And then you use this data, and this data also has the disease measurements, and you use this data to calibrate and make your models essentially reduce their bias, right? And this is a magical data, right? But it exists. And in fact, if you read all the biobank statements within the US, like, our biobank at UCLA, our former biobank at UCLA, our biobank here at Penn, other biobanks, one of their first missions there is to provide a representation of the patient population in that particular medical system, right? So those biobanks already exist. Here's a snapshot of a couple of them. This is the data I showed you before. Somewhere here, there you go. There's Penn Medicine over there. And this the chart here is looking at genetic ancestries, but you can essentially recolor them based on whatever context that you would want to do. And hopefully the big assumption here is that this sample of Penn medicine is representative of all the patient population. So once it is representative, which is a big assumption, but bear with me, if it's representative of the patient population that you're going to see, then you can use that data to calibrate all these models. Okay. So what do I mean by calibration? What I mean by calibration is that you provide the standard errors, and those standard errors contain essentially, let's say, up to 90% interval contain 90% of all the true realization of that particular prediction, right? So let's say you want to predict LDL, you predict LDL plus minus 20 milligrams per deciliter, and you're making sure that the true value sits within that range with some measure of confidence, 90%, right? And you don't want those ranges to be too high because then they're not useful, and you don't want them to be too narrow because then you're making false positives, right? And the key here is that because all the things that I showed you so far, and this is where, hopefully I can provide a more direct answer to Sarah's question from last year, these intervals have to vary by context. So, you can think about it as, let's say, in this case, it would be like, let's say, individuals of more African-American ancestry, this individuals of more European ancestry. You're more precise here because the training data comes, right? Or if, let's say, you predict LDL, these are younger folks, these are older folks, because you're more accurate there than there. Make sense? So the confidence intervals will vary by context, with the context that gives you higher accuracy corresponds to more precise or narrower bands, right? How do we estimate those? Turns out that it's actually a simpler problem than polygenic risk scores, and all that you have to do is to predict into data, let's say Penn biobank. Let's say I'm showing you what happens for LDL. You predict them across, and this is, let's say, 2 contexts. You're making more errors here, you're more precise here. So then under some statistical model, that corresponds to like larger standard errors. So you predict in lots of data, and then you literally just check how many mistakes you're making, and then you readjust the standard errors, to fit whatever to fit this 90% that you would want to do. That's how it works. It's not too complicated. I'll show you more about it if you have interest. But at the end, I think that this is essentially the performance of a polygenic risk score across genetic ancestry, across age, across sex, and you combine them into a plot like this that says these combinations of genetic ancestries, this combination of these 3 contexts give you more precise accuracy. So this is like the narrower bands, and those ones are the larger. So these are individuals where you're super accurate. These are individuals that you're not as accurate. These individuals tend to be somewhere here, so there'll be more towards, closer to the BC space, younger and more female. That's where they are, and so on and so forth. So now you've essentially made sure that all of your patients can benefit from your predictions, with the idea that you're making essentially you have larger standard errors for individuals that are further away.
How's that helping them? The standard error is super high.
Yes, so that's the next part. So you cannot really help them unless you kind of reduce the standard errors by getting more data. Right? But as a first step, you're not removing them from all of the studies. So that's the first step that I would say. That this gives you a framework where everyone can be included. But obviously, you cannot really do a good job of prediction if you haven't sampled that context or that particular ancestry. The equivalence there would be, you cannot really do a great job as much of the fancy statistical or population genetics models you might have. If you sample only Europeans, you're not going to do a pretty good job of predicting in, let's say, Africans, unless you sample.
But we knew that, right? I mean, we knew that. Of course, if you do the training in a population more similar to who you're going to apply it to, it's going to do better, right? But so are you proposing then, say, at the Penn Biobank, that you would redo also your GWAS? Like, would you redo, so, start from scratch, sort of, right? Could you also try to infer environmental covariables that would help with your?
Yes, so there are a bunch of things here. So one is to obviously these are the contexts that you can easily pull down on a piece of paper and come up, but there could be other contexts that are harder to measure. So the first part will be the figuring out what are the contexts that matter. Yeah, I suspect that every biobank will have their own context element. So that's the first one. The second one, yes, would love to redo the GWAS or the inference in the biobanks themselves, but unfortunately they're much smaller. So I think you'll lose
Exactly, that's
you lose a lot of
right. I don't know. You haven't quite convinced me yet.
I wasn't I wasn't going for it in like an hour, but this gives you a sense. If you now take this exercise and like do it across all the different traits that you have available, this gives you a sense of, and you look at the top versus the bottom, this gives you a sense what are the traits that might be more amenable to, where there's less variance. Those are the traits where context matters less, so those might be more readily for implementation. And you see things like blood pressure, things that are more closer to biology, if you will, closer to meaningful biology. Whereas on the other side, you might see things that are very complex, like BMI, everything impacts BMI. We might see like things that are out there, like education years, where the gap is really big, which makes a lot of sense based on what I told you so far, but also tells you that having a good phenotype in mind is actually part of the puzzle too. So one answer to Sarah's question might be, think about the phenotype that you might actually be more meaningful in the clinic and more meaningful biologically, because for that these contexts might matter less, and that might closer to this. So I'll conclude here by just highlighting some of the recent work for that these guys in the back of my group have done together with Ravi, showcasing how you might actually solve this puzzle. So this is the puzzle I started with from Scott and colleagues, Sarah Abramovitz, the first author of this here at Penn. Here's how it looks if you essentially now you stratify individuals, all the same data, if you will, and now you look at individuals that are fully on this side, their entire credible intervals sits in the high risk category. This is individuals that are only part of it, and this is somewhere in the intermediate. And now on the y-axis, you plot essentially the concordance of all these scores. And you can already start seeing that if their credible intervals fully sits on the top, then the concordance is pretty high, right? So it's essentially saying that we can predict what are the people that all these polygenic risk scores will agree.
Right, but are all the European ancestry people going to be on that side and ancestry going to be on that side? So then we're still doing a poor job.
Yes, in this particular case, all of these are Europeans. So in this particular case, everything is European, just about it. I'm not going to dive too much into it, but yes, you're right. So it will be very interesting to look at what's the characteristics of those, thousands of people where we can predict with statistics that they'll be highly concordant across all the polygenic risk groups. So we don't have to like throw everything out. It's just a matter of like being careful about statistics.
Or at least being more accurate about what you can tell the patient.
Yeah,
Right?
yeah.
With confidence. So that's something at least. We want to say that.
Yeah, exactly. And this is all coming from the mantra that these standard errors have a meaning attached. We're not just producing this statistical construct that are kind of not important. Stop here, take questions. I'm not going to go through it. I think I hit all these points before. So go ahead. That's
Sorry, if you want to continue.
no, I'm good. Maybe I'll just post, folks that have done a lot of this work and you've met folks here and then. Yes, go ahead.
So where does family history play in improving zero?
Family history. So you can think about it as another independent factor. So when we try to implement this in the clinic, we have 4 bins essentially in this genetically informed risk assessment score. One of them is family history. The other one is polygenic score. The other one is monogenic score. And the third one is comorbidity. These are like the 4 bins. And typically what the big studies are suggesting is to provide all the 4 of them, like are you high risk in any of them, provide all that information to a clinician, and the clinician will make their decision. So they're treated as independent. In this particular case, we can actually look at them as context. We can look at family history as a context that could differentiate the performance of polygenic risk groups. That being said, we haven't done it because data is not as easily accessible to us. But most of the stuff that I've seen, it's essentially treating them as completely independent sources of risk, which I don't know if that's truly, or it's definitely not the perfect one, or it's just a matter of limitation on data. Yes.
So with the push for more diversity in large G loss of these traits, there are some traits that have ancestry-specific results over more than just European populations. You could construct polynomial scores for more than one of these clusters.
Mhm.
How would you incorporate that data, like more than one set of ancestry-specific?
Yeah. So there are two things that I would say there. So one thing is when you think about ancestor-specific effect sizes, you're thinking about a variant that has a different frequency in multiple populations. So in other words, if someone has that mutation, it will still have the same effect size. It's just that people of this ancestry have more of those mutations, so the prevalence of that risk allele is higher there than others. So if that's the ancestor-specific type of effect, those are already incorporated in all this. In fact, the linearity of this it's kind of underlined by small changes in allele frequencies that are happening throughout. But if you're thinking about essentially G by G or G by E, where a particular allele has different effects in multiple ancestry groups, then there are ways to incorporate, but we have to find them. And I think we have to. I mean, we found a few of them, but it's not as clear and as nice to incorporate into this type of framework. So we have to think about different types of frameworks to add those types of interactions into.
Is that where local ancestry could come in potentially?
Local ancestry could come
Local
into that.
ancestry and somehow take that into account for each of these variants. It shows
Yep.
a different.
Local ancestry could definitely take it account for some of them, but not all of them. So local ancestry is kind of a local effect. So you're missing out, let's say G by, let's say global type of effect. But within 1 megabase or 2, you can account for most of this local G by local ancestry. Or maybe you have multiple causals and stuff. So there's a lot of excitement in that space. There are a lot of methods for doing all sorts of like exciting predictions. But I think most of them, they stop at this, we're gonna provide an R square, and they don't think about this thinking about standard errors on the prediction.
If you are going for clinical application, would it just be similar to develop a separate risk line and then the measure
There are a bunch of papers that are proposing to do exactly.
Including some of us from our group, where you have like as many polygenic risk scores as they are. They each, you sample different parts of the world and then you mix them based on some weights. There's some caveats to that type of approach. Every time that you're dividing the data in multiple bins, you're losing a lot of statistical power, and then you're not gaining it back by combining. So if you have all the data at once, probably the most powerful way is to run a GWAS over everything, and that will give you most of the power back. Yeah, there's a lot of there are a lot of methods in that. That's interesting. I think there was a question there.
Are effect sizes consistent across different biological contexts?
The effect size is consistent across contexts. It's a tough question, or it's a great question, but I don't think we have a lot of, to a large extent, yes. So to a large extent, we've done the local ancestry bit to look at one of the concepts being genetic ancestry. So we've looked at African-American individuals, and you can pull per individual, and you can ask within the same cell, within the same individual, whether the alleles have the same effect or not. And you're automatically correcting for all the environment, which is like one of the most powerful type of a design that we can do. And in that construct, you see very highly similar causal effects for ancestors, like to the order like they're 0.9. So you just line them up, they're perfect. And I'm talking not the frequency, just the allelic effect. However, if you take that and look at, let's say, outside of admixed individuals, like let's say Europeans versus Africans, you see much smaller correlation in defects, which could be due to the fact that there's like G-body. So I don't have more insights than that. We're observing it. We're trying to model it and see whether we can do something about it, or we can map, what are the environments that might do. It's complicated because differential environment itself without any G/E could also create, because the genetics environment, and if you change the environment part, that will also induce in this model a differential of observation. So it's a hard question, but it's a fun question, which I don't know if we have a good answer right now.
I really like the frame of this like uncertainty, just technical uncertainty for individual prediction. And I completely there's a lot of uncertainty. But there are kind of two conceptual questions. One is in your modeling of the context specific like uncertainty or uncertainty given different context. Again, it's a tagging. Right? So ultimately it's tagging some sort of environments, and depending on how far or how closely the tagging variable with the actual causal variable. If you look at an out of sample, new sample, then you are going to end up in the same problem as going to a different ancestry group, right?
Yep.
So even if you predict the uncertainty given this biobank, it may well be that if you go to another biobank, the uncertainty no longer no longer holds, right? And then so does that mean like we should spend more effort on finding the causal variant, finding the causal like environment? So that's one question. Like how much how much use is to estimate precisely the outcome versus finding the true causal things. And the second thing is, given the framework is framing like as there's a true breeding value or true genetic value for each individual in the population, and there's certain ordering, ranking, right? And then, but chances are there's prevalent G/E interaction that even the ranking may differ in a different environment, or even the extreme outliers may be just completely a different set of individuals. So, like in that case, like how do we think about like this uncertainty?
Your questions are fantastic. I think they relate a little bit to your question as well. Yes, I fully agree with you on both of them. So the first one, absolutely. So once we have the causal model, we're ready to go. So there's no, a lot of these problems go away. My take on it is that doing a lot of work in fine mapping in GWASs, and we know how far those things have become. We still have very limited evidence of actual the causal mechanisms for GWASs. I suspect it's going to be same thing with the environmental part. I suspect it's going to take a long time till we map causally, the G/E. We get insights left and right, but that's probably decades in the making. Whereas so if we have the causal mechanism, things are simple, and we can propagate a lot of things. And that will also automatically says that these credible or this calibration will vary across different biobanks, right? So the calibration will vary if we do it here, Penn, whether if we do it at ECL. It might be that the context that drive most of the calibration are the same, age, sex, and then socioeconomic, and so on and so forth. But the relative weighting in the models might vary across different biobanks. And that's definitely something that it's not perfect. It's not nice, right? Anyways, it's unpleasant, if you will, because you have to do this calibration across all of them.
Somebody moves to Philadelphia, and now their accuracy is like.
No, they're high risk. No, they're high risk. There's interaction with the cheesesteaks, if you will. But that's one of the risks that I'm willing to take on, in the sense of, you have two paths. Either wait for 10 years or so to actually get the causal mechanism done properly and then move it forward, or try to do right now something as best that we could do. And I think the best we can do is definitely not what we're doing right now, which is just give, polygenic risk scores that just give numbers to people that don't have no interpretation. It's provide them some credible intervals.
But who gets that credible interval? The patient, the doctor? Because I'm just scared, like, I don't know what I would do with my credible interval. Like, most patients are not gonna know how to interpret that. And so you're still at risk, or the doctors, if they're not variable trained on how to interpret.
There's a big training
Then-
component of it, yes, yes.
You're at
that's
risk
right.
that they're gonna start giving them medicines unnecessarily, or there could be, other consequences.
Yeah, so there's
Or
a big,
insurance,
there's a big training component that comes, so all these will be provided, in my mind, to the physician, the doctor, with proper amount of training. And I do think that there's a big room for the training in the medical schools to be done on top of, or this component needs to be propagated in the training part of it. But in general, all boils down to, eat healthy, have a good diet and so on and so forth. So the standard recommendation will still apply in general. Yeah, we're talking about just the details of the distribution. But getting back to your question, I don't think there's a general rule for all the traits, all the disease. This has to be piloted with a clinical kind of endpoint in mind. So heart disease is perfect, statins, right? That's kind of what statins are like, very, mitigated by most people. So you have the clinical endpoint, so you know what you're kind of optimizing for. Breast cancer, you have two decision points. One decision, doing essentially screening more often, or you go all the way to a surgery to mastectomy, right? So there is a clinical endpoint all the way at the end, and it's already in clinical practice. So all these polygenic risk scores will just fit on top of it if there's a clinical trial to showcase that they have utility.
Although I would argue that until we actually do these studies, it's gonna still be a lot of disparity, because you're gonna have people with certain ancestries who are just not gonna have the confidence, and they're not gonna be able to get that extra screening or whatever, so.
Yeah.
But it's a step in the right direction, but but definitely we still need to do those studies and.
Fully agree. Yes, we're not going to magically solve all the inequities and all the disparities with some statistics. Absolutely, yes, yeah. As exciting that may be. So all these will kind of like have different layers into the pie, I think.
Confidence intervals, so you kind of argue where to go next, which ones we want to shrink the most.
Mm-hmm.
All right, well, I just want to say thank you so much. This is part of a seminar series.